In this series, we are going to talk about the structure of bacteria. And of course, these bacteria fall under kingdom Prokaryote, as we already know. So bacteria are actually under kingdom Prokaryote. And of course, they have some characteristics and features which we have mentioned in the last series, especially some of the features like the genetic material they have, maybe what they use in respiration, and other criteria. But in this series, I'm going to talk mainly about the structure of bacteria, which fall under kingdom Prokaryote. First, I'm going to begin with its cell wall. And looking at the cell wall of bacteria, first, what you should note is the fact that the bacteria cell wall is very rigid. That is, it is very strengthened, especially by the presence of polysaccharide and amino acid. First, I want to look at the rigidity of the cell wall of bacteria. The cell wall of bacteria is rigid, and of course, this rigidity or this strength of the cell wall is due to the presence of murein. And this murein is a molecule which consists of polysaccharide and amino acid. So if you are being asked what exactly is the constituent or what are the materials that give the rigidity or the strength of bacterial cell, you're going to state that polysaccharide and amino acids are those constituents which is responsible for the rigidity or the strength of the cell wall of bacteria. And the function of this cell wall is to prevent the cell from bursting out, especially when it absorbs water by the process of osmosis. Now, at this juncture, I would like you to be aware that bacteria are grouped under two groups. That is, they are grouped under the aspect of the fact that there are gram-positive bacteria or gram-negative bacteria. And if you are being asked to give the difference between gram-negative and gram-positive bacteria, what you are going to state is the fact that the cell wall of gram-positive bacteria is more thicker as compared to that of gram-negative. And also, gram-positive bacteria actually have stain with the gram stain. But the gram-negative bacteria is not going to undergo any stain, especially with the gram stain. And as a result, bacteria were grouped under two different groups. We have the gram-positive bacteria and the gram-negative bacteria. Those gram-positive bacteria were those bacteria that actually undergo stain with the gram stain. But those which do not undergo any stain with the gram stain were being grouped under gram-negative bacteria. So to state again, the difference between gram-positive and gram-negative bacteria is the fact that gram-positive bacteria actually have a cell wall which is thicker, but that of the gram-negative bacteria, the cell wall is thin. It's not as thick as that of gram-positive bacteria cell wall. Also, the gram-positive bacteria actually undergo stain with the gram stain, but the gram-negative bacteria will not undergo any stain with the gram stain. Also, another extra point you should take note of is the fact that bacteria itself have a lot of environment which it occupies. It can be found in water. It can be found also in the dust. It can be found in several environments, and some of them also can be found in environment like humans, me and you. That is why at some point humans sometimes get affected by bacterial infection. And these bacterial infections sometimes can actually be eliminated with the use of antibiotics such as penicillin and other drug substances. And what you should note is the fact that when these bacteria found themselves in hosts such as animals or humans, then of course they must try every possibility to protect themselves from antibacterial enzymes being produced by humans, which are like the host for these bacteria. For example, when these bacteria found themselves in humans like us, what happened is the cell wall of these bacteria will be threatened by antibacterial enzymes being secreted by tears, saliva, and other body fluid. So when the body produces saliva or these enzymes like lysosomes from tears and saliva, what happens is it goes ahead to try to digest the cell wall of bacteria. And these antibacterial enzyme being secreted by the body, of course, will try to attack the cell wall of these bacteria and causes the digestion of the polysaccharide, which is the major constituent that gives the strength of the cell wall. Of course, I've earlier mentioned that the cell wall itself is being rigid due to the presence of murine. This murine consists of polysaccharide and amino acid as well as lipid. So what happens is when these antibacterial enzymes are being secreted by the body, it goes directly to try to destroy the cell wall of bacteria, especially when they find themselves in environment like humans or animals. So the polysaccharide of the cell wall of this bacteria will be digested and causes the cell wall to become weakened. And when the cell wall becomes weakened, what happens is when the bacteria now absorb water by osmosis, it is going to cause the cell wall to burst and it's going to cause destruction of the bacteria itself. This same logic applies in medicine. Of course, for example, when a patient is being attacked by bacterial infection, of course, what happens is these patients are being prescribed with antibacterial drugs. These antibacterial drug, when it is being taken by humans, what happens is it poses an effect on the cell wall of the bacterial cell. And once this effect is being posed on the cell wall, what happens is these drugs being given or prescribed to patients, especially penicillin and other antibacterial drugs, it goes directly to the cell wall of this bacteria to see how it can weaken the cell wall by trying to digest the polysaccharide as well as digesting other nutrients which make up the cell wall. And once these constituents are being digested or destroyed, what happens is the cell wall becomes weakened. And when this bacteria try to feed from its host by absorbing nutrients or water from its host, since the cell wall has become weakened, what happens is there will be bursting of the cell wall. And as a result, it is going to lead to death or elimination of this bacteria existence within its host. It should be noted that all the constituents or the materials within the bacterial cell is being surrounded by a partially permeable membrane. And looking at the membrane of bacterial cell, there are three major, very important membranes we should take into consideration, which of course the cell wall start to have this infolding of the membrane, and there are three major membranes I'll list here. First, I want to look at the photosynthetic membrane. At this level, it is an area or location where photosynthesis actually takes place. So this photosynthetic membrane possesses some photosynthetic pigment. It is at this level where photosynthesis actually occurs within the cell of the bacteria. Next, we have mesosomes. It is also an infolding of the cell surface membrane that forms the mesosomes itself. The mesosome is a membrane that has a lot of function. The first function of the mesosome is the fact that it helps in positioning the DNA of the bacterial cell in times of cell division. Because during the time of cell division, what happens is the DNA of the parent cell must undergo replication. And for this process to take place, the DNA itself must be put in a good position for it to be in place. And what helps in positioning this DNA is the mesosomes. It tries to attach itself to the DNA and put it in place for replication to take place, especially in the process of reproduction. In other words, one of the functions of the mesosome is to help to position the DNA for it to undergo cell division during the process of reproduction or during the process of cell division. The mesosomes also play another role in the formation of the new cell wall of the daughter cells during cell division. Of course, when cell division actually takes place, what happens is there is replication of the parent cell to form two identical daughter cells, especially in the asexual reproduction. And when this happens, what happens is when the two daughter cells have been formed or replication has been completed, what happens is there need to be the new formation of cell wall for this daughter cell. And what plays this role is the mesosomes will help in the formation of the new cell wall. So these are some of the functions of the mesosomes. And thirdly, the mesosomes play a role as the respiratory site for the bacterial cell. What happens is when you talked about the respiratory site, this is the area where it aids the bacteria in respiration. So first, we have talked about differences between prokaryote and eukaryote, and most organisms under prokaryote are bacteria. And when we talked about the difference in terms of respiration, for prokaryote, what they use in respiration is mesosomes. But for eukaryote, they use mitochondria for aerobic respiration. So I've listed 3 major roles of the mesosomes when it comes to the cell membrane of these. So I'm going to end here for today, and we are going to continue with the next series based on the structure of the cell wall. So we have mentioned the cell wall and the cell membranes in this series. In the next, we are going to talk about other structure of bacterial cell. Also, make sure to check the quizzes we have set up for you to answer some of the questions based on this series to keep yourself up to date. You also want to make sure that if you have not yet received this quiz, request for it below this video description, and we are going to make sure that we give it to you to see how you can test or quiz yourself based on this particular series to keep yourself up to standard. Goodbye for now, and see you again in the next series.